Hello, in this video lesson we will talk about high performance liquid chromatography. Sometimes it is written as high pressure liquid chromatography. Both of them are considered to be true because high performance liquid chromatography uses high pressure. Okay, high performance liquid chromatography, further I will call HPLC. Has, let's say, this kind of work mechanism. Let's say it looks something like that, and it has solvent reservoir, pump, injector, HPLC column. It has detector and the software system, including the monitor and processing system, and the waste liquid is gathered in one bottle like this. The main function of the HPLC is to separate the compounds in the given samples and quantify based on the retention time. The compounds can be identified, and based on the peaks, the compounds can be quantified. So we will talk about how it works in HPLC. Here we can see uh, how it looks, how the modern HPLC looks. It has this kind of sections. The ups one can belong to the pop, and then injector, and the column holding part, degassing part, and other parts. Here, let's take a look how it can work. From the solvent reservoirs, at least they are expected to be two. These samples are taken by using the vacuum pump. Okay, in isocratic, there could be one pump. In gradient regimes, gradient elements. Okay, two pumps are expected, or the pumps that might take the samples from two pumps is provided, and then they are mixed under the high pressure. Okay, they can be pre-column, let's say degasser, for example, yeah, and then they are given to the analytical column by the together with the studied sample. They are sent to the analytical column, and in the analytical column they are separated. I mean, the compounds in the studied sample are separated from each other while they are subjected to sorbent, something like silica gel holding octadecyl derivatives. And when the compounds are separated from each other, they are identified and quantified by using different detectors. We are going to talk about detectors a bit later, and other tools a bit later. So the after detection and quantification, the result is are, let's say, studied by using different softwares. So this is how the HPLC is utilized by an operator. And by this text, we are going to understand more in detail about the principles of how HPLC works. The separation principle of HPLC works on the distribution of samples between a mobile phase and eluent and the stationary phase packed on the column. And about the mobile phase and stationary phase, I recommend you to know from our other presentations. HPLC chromatographic peaks in the elution curve are affected mainly by flow rate, particle size, diffusion rate, and thickness of the stationary phase. So Again, the main mechanism of identification by using HPLC is based on the retention time of the exact compound in the exact condition, including the exact flow rate, the exact solvent, the exact ratio of the solvent, and so on. So the retention time is expected to be standard for the same compound for the same condition. So based on that, the compounds can be identified compared by comparing to the retention time of the standard, let's say, compound. And these processes can be affected by many factors, including flow rate, particle size, diffusion rate, thickness of the stationary phase, and others. Uh, and high performance liquid chromatography principle solved all the problems of classical methods of separation, let's say. So the HPLC allows us to identify the compounds based on the retention time comparison to the standard compounds, and then quantify them. Quantification of compounds by using HPLC is also possible. And therefore, the problems that belong to former approaches of column chromatography, let's say, were almost solved in an example of HPLC. From the point of view of speed and simplicity, gas liquid chromatography system is better than the HPLC system from the point of view of speed and simplicity. However, for the isolation of non-volatile substances, including inorganic ions or thermally unstable materials, the HPLC system is better anyway. So the gas liquid chromatography has its own, let's say, advantages such as speed, yeah, or simplicity. And the HPLC system, liquid chromatography system, has even has its own advantages, like the using possibility for non-volatile substances as well. Okay, here we can see again the pump takes the solvent, sends to the injector. To the injector, we can send the sample, or by using the auto sampler, yeah, and the injector sends the mixture of the sample together with the solvent to the column. In the column, the separation happens, and then based on the volume of the peaks, we can quantify the compound, and based on the retention time, we can identify the compound. So the HPLC has its own software, and by using its own software, we can identify and quantify the compounds. Next. Okay, HPLC principle. We are going to read and understand the text. The purification takes place in a separation column between a stationary and mobile phase. We talked about this a bit. The stationary phase is a granular material with very small porous particles in a separation column. If the sorbent is smaller, okay, it will be easier for us to separate the compounds in the column. The mobile phase, on the other hand, is a solvent or solvent mixture that's forced at a high pressure through the separation column. Uh, we are required to optimize the condition. The optimization of the condition requires identifying the solvent which can be effectively used to separate the compounds with similar structures. So the mobile phase and its components, and whether they are mixture, if they are mixture, the ratio of solvents are very significant in order to achieve higher efficacy of HPLC utilization. Via a valve with a connected sample loop, that's a small tube or a capillary made of stainless steel, the sample is injected into the mobile phase flow from the pump to the separation column using a syringe. Again, in older versions, syringes were used to the to send the samples. And today, up to my understanding, many HPLC tools are equipped with the auto sampler. And if you want to buy the cheaper one, okay, it can be possible to find the HPLC equipped with the syringe linked injector. And subsequently, the individual components of the sample migrate through the column at different rates because they are retained to a varying degree by interaction with the stationary phase. Okay, in the stationary phase, we know there is liquid flowing with the constant flow rate, and there are the compounds there to be separated. Based on the interaction of the samples with the silica gel or its derivatives like C18. So these are the molecular bases that provide the separation of the different compounds in the HPLC column. After leaving the column, the individual compounds are detected by a suitable detector and passed on as a signal to the HPLC software or on the computer. We are going to talk about the types of detectors. And HPLC are equipped with different detectors such as UV-Vis detector or diode array detectors, fluorescence detectors, or it can be HPLC can be integrated to mass spectrometers. Mass spectrometrically, some compounds can be identified as well. At the end of this operation or run, a chromatogram in the HPLC software on the computer is obtained. Every single HPLC has its own software, and they are, the HPLC are provided with their own software, and that is the only way how we can effectively use the HPLC. If there is some problem in the software, okay, it's almost impossible to use those HPLC effectively. The chromatogram allows the identification and quantification of the different compounds. In the chromatograms, we can find out the retention time. So based on the retention time, we can suggest the compound compared to the standard compounds. And based on the peaks, we can quantify the identified compound. Okay, next, pump. Pumps. Now we are going to talk about the components of the HPLC. Pump is one of the most significant, let's say, components of the HPLC. And in this website, okay, approximate prices of the pumps are given here. The development of HPLC led to the development of the pump system. Okay, now we have updated highly precise HPLC because they are equipped with highly precise pumps as well. The pump is positioned in the most upper stream of the liquid chromatography system and generates a flow of fluid from the solvent reservoir into the system. And high pressure generation is a standard requirement of pumps. Besides this, it should also be able to provide a consistent pressure at any condition and a controllable and reproducible flow rate. As I reminded earlier, pump is very significant because again, we can identify the compounds by comparing the retention time of the studied compound by comparing it with the standard one. So the exact, let's say, pressure for the liquid for the flow rate should be provided. Otherwise, okay, if there is a difference between the flow rates of the studied sample and the standards, we can witness some differences, and those differences might cause some complexity in the study process. Most pumps are used in current LC systems in liquid chromatography systems, which generate the flow by back and forth motion of a motor-driven piston. And because of this piston motion, it produces pulses. Okay, in liquid chromatography, in the classical liquid chromatography systems, we could see different sending pumps like peristaltic pumps, yeah, and the system of the HPLC is very precise. They are not comparable with those ones. They work very precisely. That is why we can rely on the precision of the HPLC. And injectors are the second most, let's say, significant components of the HPLC. By using the injector, we can send these samples as indicated in these figures. By using some syringes like this, we can take some exact amount of the liquid from 10 to 50, let's say, micrometer. So then we can send it to the injector. The simplest method is to use a syringe, and the sample is introduced to the flow of eluent. I have to highlight that this kind of manual sending of these samples were provided to the HPLC produced, let's say, at least 10 years ago, yeah, until 2010, 2015, yeah, for example. And today's almost all HPLCs are provided with the auto samplers as indicated here. So these places are filled with the vials as indicated here, brown colored, protected from the sunlight. So inside here we can see the automatic auto sampler, something like this, have their own syringe that can take some amount from the vials that could be numbered, that could be given to the order. Yeah. So then from the exact vial, they take some samples and send it to injector that we call auto injector. And this is how the samples are taken and sent to the injector. Okay, and now we are going to discuss some info about the columns. Okay, again, columns are filled with the sorbent like silica gel or silica gel C18 derivatives, as indicated here. Silica gels are linked with octa diethyl derivatives. So then they are provided with the ability to hold some compounds with higher hydrophobic nature for a longer time. Okay, this is why silica gel is provided with C18 tails. Okay, they can be utilized the nonpolar interactions for solid and to provide more nonpolar interactions for solid phase extraction. Okay, the column is used for the separation. Again, the recent columns are often prepared in a stainless steel housing instead of glass columns because the pressure inside is very high. The glass ones may not be stable enough to hold the high pressure inside it. And packing material generally used is silica or polymer gels compared to calcium carbonate. The eluent used for liquid chromatography varies from acidic to basic solvents. Most column housing is made of stainless steel since it's tolerant to a large variety of solvents as well. I would like to remind you that strong acids like sulfuric acid or hydrochloric acids are usually not used for HPLC because they are highly acidic, and those highly acidic condition might cause the breakage of covalent bonds like this, so they can disrupt the sorbent. That's why if we want to provide the acidic condition, the acidic condition to be provided can be the kind of weak acidic condition. So in some cases, some acids like phosphoric acid is used, but not sulfuric or hydrochloric acid. Okay, next. Detectors. Again, detectors can be different. They are used to identify and quantify the compounds based on the retention time and flow rate. And separation of analytes is performed inside the column, whereas a detector is used to observe the obtained separation. The composition of the eluent is consistent with no analyte is present. While the presence of the analyte changes the composition of the element, uh, what the detector does is to measure these differences. Okay, the composition again can be defined by using the detectors. What detectors do is, okay, they quantify the compound, and the identification is, let's say, done by based on the flow rate and retention time. Okay, this difference is monitored as a form of an electronic signal. There are different types of detectors as indicated here. The simplest one, the most widely used one is UV-vis, ultraviolet visible detectors. They measure the amount of light absorbed by sample at a specific wavelength. We have special Okay, we have separate video lesson about UV-V spectroscopy. UV-V detectors are best for compounds with chromophore groups. Yeah, if the compounds have their own chromophore groups, they can be identified, quantified by using UV-Vs. If not, okay, those compounds are to be reacted with chromophore groups, and the quantity of those chromophore group holding derivatives is identified. So then quantification of those compounds not holding chromophore groups can be carried out. And the advantage of these detectors are they are stable, they are sensitive and relatively inexpensive compared others like DAD, diode array detectors. They can also be termed as photodiode array. And they are a more advanced version of the UV detectors that can scan a whole range of the wavelengths simultaneously. At the same time, by using DAD, it's possible to find out to carry out the quantification of various compounds. And they are best for identifying unknown compounds and checking peak purity. And the advantage of them is that they provide a 3D profile, including time, intensity, and wavelength. And another one is refractive index detector. The compounds that do not have chromophore groups like sugars, carbohydrates, other carbohydrates, alcohol, or polymers can be detected by using refractive index detectors. Yeah, and its disadvantage is that they are not very sensitive and cannot be used with the gradient illusion. We are going to talk about the gradient and isocratic regimes a bit later. And however, they are best for compounds that do not absorb UV light. Sugars, again, alcohols, polymers can be identified with refractive index detector. And the next type is fluorescence detectors. Uh, we have a separate topic about the fluorescence, a separate video lesson. I recommend you to watch those video lessons again. And in fluorescence process, okay, some light is given, and those light can be emitted with the higher wavelength with smaller photon energy. So it is possible to use to detect those compounds based on the emission coming out from those compounds. And they are best for naturally fluorescent compounds like vitamins or environmental pollutants. Yeah. And the advantage of the fluorescence detectors is that they are extremely sensitive, up to 1,000 times more sensitive compared to the UV detectors. So with the concentration of some compounds that have the fluorescent property, and they are given at 1,000 times smaller concentration, it is still possible to quantify them. Okay, recorder. The software system is linked with these. I mean, the detector identify the let's say the signals and send to the recorder. The change in element detected by a detector is in the form of an electronic signal, and thus it is still not visible to our eyes. In older days, the pen paper chart recorder was popularly used. You might have seen those kind of paper-based ones, and today all these software are provided with the operational system, provided with the display. Yeah. Nowadays, a computer-based data processor, yeah, termed integrator, is more common. There are various types of data processors, from a simple system consisting of the inbuilt printer and word processor to those with software that are specifically designed for an liquid chromatography system. And they include not only data acquisition, but also features like peak fitting, baseline correction, automatic concentration calculation, molecular weight determination, if they are linked to mass spectrometers. Yeah, so these features can also be analyzed in recorder or in total in the software of the simple of the tool. The gasser is another part that is very significant. The gassers, here we can see that the eluent going through the pump to the column is degassed by using a tool termed degasser. This is used for the eluent to be degassed. The eluent used for liquid chromatography analysis may contain gases such as oxygen. It's not visible to our eyes. Yeah, when the gas is present in the eluent, it can be detected as noise, or it might cause an unstable baseline. So the degasser is very significant. It uses a special polymer membrane tubing to remove gases that could be present in liquids. The numerous very small pores of the surface of the polymer tube allow the air to go through while preventing any liquid to go through the pore. So degasser is another significant point that should be provided to the modern HPLC tools. And column heater, there are special requirements for the HPLC. For example, the rooms where HPLC are used should be provided with the constant temperature, let's say 25 centigrade or 30 centigrade. For any case, for some cases like, okay, there could be some kind of deviation in the temperature that could affect the retention time or the viscosity of the eluent solvent. Yeah, and there could be kind of discrepancies in the retention time of the samples. That is why, okay, columns are provided with the column heater or column chilling, let's say, tools. column chilling or column heating tools. And another significant and useful point of the column heater, not all compounds, for example, can be identified by using HPLC in a room temperature, at a room temperature. At a room temperature, for example, at 25 or 30 centigrade, the identification of some compound is that might have high viscosity may not be provided, may not be appropriate. So the temperature necessarily could be increased up to 50 or 80 centigrade in some cases. Therefore, the column heaters are very useful for those cases. Okay, next. Here we can see the significance of the column length. Let's say if we have two different compounds, black and dark blue and red, and they have to be separated. If the column is not long enough, okay, we may not see some exact separation of A and B compounds, red and blue compounds that could be detected with the peaks, something like this. Okay, and so these are, let's say, well-resolved compounds. Resolution is an important HPLC performance indicator, usually assessed by how quickly and how completely target components in a sample separate as they pass through a column. Resolution is measured by dividing the difference in peak retention times, by the average peak. With resolution can also be expressed in the resolution equation as a combination of the factor separation efficiency and retention that affect this value. And here are the factors that might affect proper resolution, proper separation of the compounds in the column. Okay, the factors that might affect this process can be the increased column length, as I mentioned earlier, and the decreased particle size. If the particle size of the sorbent is smaller, okay, we can expect a more precise, let's say, it will be slower, but it will be more precise separation of red and blue compounds. And reducing pigtail is another factor. Increasing the temperature can be another factor. Okay, we talked about the significance of the temperature. The temperature is especially significant for the compounds that might have high viscosity. The reducing system extra column volume is another point that might significantly affect the resolution. And the column stationary phase is another point. The pH of the mobile phase and the solvents of the mobile phase are two other points that might change the selectivity. And the increase of the retention can be provided by using a weaker solvent. We can provide it by changing the polarity of the solvents and by changing the ionization, which means that by changing the polarity of the analyte, by changing the pH, and using a stronger stationary phase that could be provided by changing the polarity, could be another reason to increase the retention time. So these are about the separation in the column. And here we can see analysis of total amino acids in rice using a validated reverse phase high performance liquid chromatography method with diode array detection. And we talked about different types of detectors, and diode array detector is a modern type of UV detector, as I earlier mentioned, and the range of these DAD can be between 190 and 950 nanometers. Unlike fixed wavelength detectors, DADs allow for multi-wavelength analysis, peak purity verification, and high sensitivity, high speed data acquisition. Here we can see the samples of amino acids that were obtained by using DAD, diode array detectors. Here we can see different amino acids, aspartic acid, glutamic acid, serine, yeah, histidine, and the ending points are lysine or leucine, something like that. And obtained, some of them are obtained at 262 nanometers, and others were obtained in 338 nanometers. So the nanometers of the wavelength, I mean the wavelength is also very significant to to see the, let's say, absorbance intensity of the compounds, which can enable to analyze them at their lower concentrations. Okay, next. And here we can see uh, uh, concentration of amino acids identified by fluorescence detector, FLD fluorescence detector, and these ones were identified DAD. It's not linked with the detector here in these two conditions. It is linked with the varying composition of the eluent. Here we can see method one, mobile phase has in a bottle 14 millimolar aqueous sodium dihydrophosphate buffer, pH adjusted to 8.5, and in B bottle, okay, there is a mixture of acetonitrile, methanol, water, and phosphoric acid at 45 to 10 to 0.1 mass ratio, volume ratio, I'm sorry, VV, volume ratio. And in the second case, here we can see the mobile phase A included ammonium formate, 60 millimolar, and pH 5.5, and B has ammonium formate, 60 millimolar, pH 7.5, and C bottle has ammonium formate 10 millimolar and formic acid 0.1 molar in acetonitrile and water with the ratio 90 to 10, 9 to 1, volume ratio. And in both cases, I think gradients were used there. Gradient. We are going to talk about gradients a bit later. The point we have to pay our attention to is that for the identification of amino acids, the authors used this compound, name it 9-fluorenyl methyl chloroformate, because it has a chromophore group, or it has the liability to emit fluorescence as well. So by providing this kind of compounds, it is possible to detect the amino acids at UV range. Let's say around 300 nanometers. Why? Because not all amino acids, okay, excluding phenylalanine, tyrosine, or threonine. Other amino acids may not be identified based on the absorbance of UV light, because other amino acids, excluding those three, phenylalanine, tyrosine, and threonine, other amino acids do not absorb UV light. So they are linked with this kind of molecules having a chromophore group or fluorescent group. And so then their quantification can be provided, they can be quantified. Okay, and another method of identifying amino acids or any other compounds is the integration of HPLC to the mass spectrometer. HPLC part is used to separate different amino acids, and mass spectrometry is used to identify the compounds based on the mass analysis of compounds by using the mass spectrometer tool. Okay, so in this article, the authors used the integrated tool HPLC plus mass spectrometry. Okay, now we are going to understand to understand more about the mobile phase. Mobile phase can be prepared by mixing different solvents like water, acetonitrile, methanol, yeah, and they are filtered before sending to the column. Usually, when the pumps are taking, they take the filtered solution, and they are degassed, and they can be sent to the column in two regimes, either isocratic or gradient. Yeah, in gradient, the composition changes over time. And here we can see the structure of acetonitrile, which is polar aprotic, and this is methanol, which is polar protic. It reduces hydrogen, yeah, releases hydrogen, and here it does not release hydrogen. So even if they are both have, let's say, medium polarity, but this one is aprotic, this one is protic. And this is about the isocratic elution. Again, in gradient, we change the concentration of the eluent. Here we can see 10% B is sent between 0 and 5 minutes in the first 5 minutes, and in the next 15 minutes from 5 to 10 20 minutes, okay, the concentration of acetonitrile, for example, is increased from 10 to 90. So this is the difference between gradient elution and isocratic elution. In isocratic elution, the composition is expected to be constant. It does not change for the whole 20 minutes. At the beginning, it was holding 70% water and 30% acetonitrile, and the ending eluent holds the same composition as well. Okay, if we have methanol and acetonitrile, okay, what is the difference that could affect the flow rate and the sequence of the compounds that could flow, that could be washed out? Or okay, the difference can be explained with this. Okay, methanol is protic and acetonitrile is aprotic, and here we have methanol and acetonitrile. Benzoic acid, the black compound, has protic nature. It releases proton as well. So by the nature, methanol and benzoic acid is expected to be closer to each other compared to the interaction acetonitrile, which is aprotic, and benzoic acid. Yeah. So if we want to separate these two compounds from each other, and if we use methanol, okay, the benzoic acid is expected to be released first, to be washed out first, to be identified first. Okay, and because it is protic, this is protic, and pKa is expected to be 4.2. Weak acid, here this pKa is expected 10, very weak acid. So if we elute the mixture of these two compounds by using acetonitrile, so the release of phenol is expected to be faster than benzoic acid. And these are the points that we have to pay our attention while we are learning the mechanisms and while we are developing the methods for the identification and quantification of some compounds. Here we can see that not only the interaction between methanol and benzoic acid and phenol, or acetonitrile, benzoic acid and phenol, but also there is the role of the interaction between the eluent and the sorbent, silica gel CAT. Yeah? These are also the pointers that affect the retention of phenol or benzoic acid that could be eluted with either methanol or acetonitrile. Next. And now this is about comparison of isocratic and gradient HPLC. By the features, we can differ them. First, isocratic means the composition is constant, it does not change. And in mobile phase, we observe, we provide the changes. For example, 70% water and 30% acetonitrile composition is kept during the whole 20 minutes of isocratic elution, as we talked earlier. And in graduate elution, the percentage of acetonitrile, for example, is increased from 10 to 90 minutes, starting from the 5th minute, ending at 20th minute. Okay, and by the nature, as we see that isocratic elution is simple, much more simple than gradient elution. And isocratic elution is best for simple mixtures with similar polarity. But if the compounds are in mixture like extractors, the gradient elution is expected to be more appropriate than the isocratic elution. And the runtime in isocratic is often longer for complex samples, because the separation in isocratic elution may not be as effective as in gradient solution. And in gradient, it is usually a bit shorter, let's say not much. And peak shape in isocratic is a kind of broad, and in gradient we can witness sharp peaks, especially for late eluting compounds. And the resolution in isocratic, it's good for simple mixtures, and gradient is better for complex mixtures. Reproducibility of isocratic elution is easier, that's why higher, and the reproducibility of the gradient elution is a bit complex and slightly lower. Yeah, and equilibration time is shorter. This is equilibration time, not run time. Run time here is longer, but equilibration time here in isocratic is shorter than the gradient one. We are going to talk about those times later. And column stress in isocratic is lower, and in gradient it's higher. Solvent consumption is lower in isocratic due to the shorter, let's say, time here. And here it's often higher, often, not all the time. And baseline stability. It is more stable in isocratic, and here we can witness some drifts during the gradient. Method development will be easier in isocratic, and in gradient elution it is more complex because the optimization is required for gradient elution. And isocratic elution can be used for routine analysis, let's say, but for the quantification and identification of different compounds in the composition of extracts, for example, gradient elution is necessarily used. So we can use gradient elution for pharmaceuticals, proteomics, complex samples, and others. Okay, here we can see we can find some info about the timing of different stages, including the preparation, running, and after running washing processes of HPLC. The first one is column setup. Column details. Okay, columns length can be between 50 to 250 millimeters. The higher the column, the more precision, the higher precision of the samples. The longer the column, the more expensive the column. And diameter can be between 2.1 and 4.6 millimeters, and the sorbent particle size is expected between 3 to 5 micrometers. And again, the smaller sorbent particle, the higher precision of the separation. And the temperature can vary. Let's say the set temperature can vary between 25 to 40 centigrade, but the lower temperature is preferred for any case. So at room temperature, carrying out HPLC analysis at 25 or 30 centigrade is preferable. But again, in some special cases for the compounds that have higher viscosity, the temperature of the column is necessarily increased up to 60, 70, or 80. Yeah. And equilibration time. Before injection, the column must be equilibrated with mobile phase. The equilibration time can last from 10 to 30 minutes. The average time can be 15 to 20 minutes. Yeah. Otherwise, okay, we may not see the expected results. Or the column can be washed with 10 column volumes of the liquid, of the eluent. An example can be like flow rate 1 milliliter per minute. This is for equilibration, 1 milliliter per minute, and column volume 2 milliliter. And it could be equilibrated for 20 minutes approximately. Yeah, let's say moderate time. Okay, and sample injection. This is equilibration time, and this is about sample injection. Sample injection volume can vary from 5 to 50 microliters, depending on the concentration of this studied compound. And it can be sent by manually or by using autosamplers. In different HPLC, different options could be provided. And here, the separation, the run phase have some significant differences in the flow rate and flow rate, let's say. Flow rate of the separation can be between 0.5 to 1.5 milliliter per minute. It might not be significantly different in those cases. Here it is flow rate of equilibration time is 1 milliliter per minute. Let's say it is similar. They could be similar as well. And if a faster preparation of the equilibration time is expected, the higher flow rate could be selected as well. And the runtime is between 5 to 30 minutes based on the retention time of the studied compound. If the compound is to be studied identified in the early minutes, like 5 to 15 minutes, okay? And if we witness the absence of other compounds in the remaining 10 minutes starting from 20 minutes, it can be possible to stop it at 20 minutes, or 5 to 20 minutes could be selected for further analysis. Okay, and what happens? Compounds interact differently with the stationary phase. They elute at different times, so they cause different retention times. And now this is about the detection. Common detectors, as we talked, could be UV detectors, fluorescence detectors, mass spectrometry, the HPLC could be integrated to the mass spectrometry, Yeah, so mass spectrometry can also be used for the identification and quantification of the compounds that are separated in the HPLC column. And column washing, this is also very important. After running the separation, the column is required to be washed, and higher percentage of acetonitrile or methanol can be used for washing purposes, which could last from 10 to 30 minutes, yeah, or 5 to 10 column volumes. What is the purpose? Again, the purpose is to remove strongly retained compounds and prevent the contamination and ghost peaks. This is very significant. After using the HPLC for the same purposes, let's say we used it 10, 15 times a day, and after utilization, they have to be washed, possibly with higher concentration of acetyl element, because the higher concentration of a more hydrophobic compounds like acetonitrile compared to water, so then the compounds that might have higher hydrophobic interactions with the C18 silica gel could be more effectively washed with higher concentration of acetonitrile. Okay, now this is about the rare equilibration. After washing, rare equilibration should also be provided, which could be carried out for 10 to 20 minutes with the same concentration of equilibration, let's say liquid, eluent, thiam, flow rate, and others. Okay, and as a summary, we can say that equilibration could last from 10 to 30 minutes. Sample run could be 5 to 30 minutes. Column washing can be 10 to 30 minutes. Rare equilibration can be 10 to 20 minutes. And an example here is, for example, if we use the column filled with C18 silica gel, with a column length 150 milliliter, and the flow rate can be 1 milliliter per minute. The temperature can be set up to 30 centigrade. The gradient of the acetonitrile can be from 10 to 90%, can be enhanced, can be increased from 10 to 90%, and the runtime can be 20 minutes. It could be washed with 90% acetonitrile for 15 minutes and could be re-equilibrated for 15 minutes with the equilibrate element system. These are all that I wanted to deliver you. If you find this info useful for you, I kindly ask you to support us with your likes on YouTube, so then our video lessons could reach a wider audience. These are all, and I express my thanks to you all. Have a good day. Bye.